ప్రిన్సిపల్ సార్ ఇప్పుడే ఐదు
Sir, shall we wait for 5 more minutes?
రిప్లై ఇచ్చేసాం కదా ఒక 2 minutes పడుతుంది.
Good afternoon, Hanmi Reddy, sir. Sir, can we wait for 5 more minutes?
Yeah, we can wait. No problem.
Okay, sir.
యూట్యూబ్ లేదు కనెక్ట్ అయ్యింది.
Am I audible?
Yes, sir, you're visible, sir.
Okay.
అమూల్య స్టార్ట్ చేసేయండి ప్రోగ్రామ్.
Okay, sir.
నేను ఫేస్ లేకుండా ఉంటే నీట్గా వస్తుంది.
Good afternoon, everyone. Today, we have assembled here virtually with a shared commitment to empower faculty excellence, as we all know that in this ever-evolving landscape of education, faculty members play a pivotal role in shaping the future of our students, institutions, and the nation as a whole. This is C.H. Amulya, and I am honored to welcome you all on behalf of Sir C.R. Reddy College of Pharmaceutical Sciences to this national level online 1-week faculty development program with the theme Empowering Faculty Excellence, Enhancing Research Competence and Innovation, organized by Sir C.R. Reddy College of Pharmaceutical Sciences. Sir C.R. Reddy College of Pharmaceutical Sciences is an esteemed institution with a rich history and a focus on academic excellence, research, and community engagement. Our college was established in 2007 with the objective to educate and innovate to meet the evolving needs of the pharmacy profession, ensuring excellence in pharmaceutical education and research. Pharmaceutical education programs offered in our college are Diploma in Pharmacy with 60 seats, Bachelor of Pharmacy program with 100 seats, PhD program with 60 seats, and Master of Pharmacy program in Pharmaceutics, Pharmacology, and Pharmaceutical Analysis and Quality Assurance departments. It is a privilege to host this virtual gathering of educators and researchers from across the nation coming together to explore and elevate the standards of academic excellence. This 1-week faculty development program is designed with the objective that all the participants will acquire the knowledge, skills, and motivation necessary to enhance their research competence, fostering a culture of innovation in their educational institutions and producing impactful scholarly work that contributes to the advancement in their respective fields. This 1-week program features highly talented and esteemed resource persons with one distinguished expert scheduled for each day, starting with day 1, Dr. B. Hanmi Reddy, sir, Director, Analytical R&D, Slayback Pharma, Hyderabad. Day 2, Professor Sheikh Afzal Basha, sir, Professor, Department of Pharmaceutical Chemistry, St. Mary's College of Pharmacy. Day 3, Dr. S. Pavan Kumar, sir, Associate Professor, Department of Pharmacology, Sri Vishnu College of Pharmacy. Day 4, Dr. Shahin Begum, Madam, Associate Professor, Department of Pharmaceutical Chemistry, IPT, Sri Padmavati Mahila Viswavidyalayam, Tirupati. Day 5, Dr. M. Murli Krishna Kumar, sir, Assistant Professor, Department of Pharmaceutical Chemistry, Andhra University College of Pharmaceutical Sciences. Day 6, Dr. A. Lakshman Rao, sir, Professor and Principal, VP Institute of Pharmaceutical Sciences, Gudlavaleru. And day 7, Professor Sheikh Abdul Rahman, sir, Dean, School of Pharmacy and Health Informatics, DIT University, Dehradun. Let us start the event with a virtual lamp lighting ceremony symbolizing the illumination of knowledge and the beginning of our journey to empower faculty excellence and foster innovation. REDDY College of Pharmaceutical Sciences is privileged to have a proactive and influential correspondent, Sri M. Raja Ram Mohan Rao sir, whose exceptional leadership and unwavering support have been the driving forces behind our institution's remarkable growth and ongoing success. I request our correspondent, sir, Sri M. Raja Ram Mohan Rao sir, to share his valuable insights.
Ladies and gentlemen, esteemed faculty and distinguished guests, I am truly honored to welcome you to the opening of our one-week online National Faculty Development Program, themed Empowering Faculty Excellence, Enhancing Research Competence and Innovation. This event marks a significant milestone in our ongoing commitment to the growth and development of our academic community. In a world that constantly evolves, the role of faculty members is more critical than ever. This program aims to empower our educators, equipping them with the knowledge, skills, and innovative approaches necessary to excel in their roles and make a lasting impact in the field of education. I would like to express my heartfelt gratitude to all the speakers, facilitators, and participants who have made this program possible. Together, we will strive to raise the bar of faculty excellence, ultimately benefiting our institutions and, most importantly, our students. Once again, welcome to the Empowering Faculty Excellence, Enhancing Research Competence and Innovation Faculty Development Program. Let us make the most of this opportunity, inspire one another, and collectively shape the future of education. Thank you, and let the learning begin.
Thank you, sir. Our college is fortunate to have a dynamic and visionary principal, Dr. A. Narendra Babu sir, whose visionary leadership and relentless commitment have been the guiding light of our institution's journey. I now request our principal, Dr. A. Narendra Babu sir, to deliver the opening remarks. Principal sir.
One minute. Is it audible?
Sir, your voice is not audible, sir.
Just a minute. So now is it
Yes,
audible,
sir.
my voice?
Yes, sir. Yes, sir.
So good afternoon, sir. So good afternoon to all. Ladies and gentlemen, esteemed colleagues and distinguished participants. So I am truly delighted to welcome you all to this one-week online National Faculty Development Program with the theme Empowering Faculty Excellence, Enhancing Research Competence and Innovation. So it is a pleasure to have such a diverse and enthusiastic group of educators and researchers gathered here to embark on this transformative journey. So the main objective of this program is to provide a platform for our faculty members and the faculty community to enhance their research competence as well as fostering innovation. So we recognize the critical role that faculty members plays in shaping the educational landscape and pushing the boundaries of knowledge. This one-week-long initiative aims to equip you all with the tools, strategies, and resources necessary to excel in your roles as educators and researchers. So during this program, you will have the opportunity to engage with expert speakers, share experiences, and collaborate with your peers. Together, we will explore various topics which are related to the research methodologies, teaching strategies, and using the technology for enhancing your teaching and research capabilities. And the main and ultimate goal of this faculty development program is to empower you all to make a lasting impact on your students and the broader academic community. So as we progress through the various sessions and activities, I encourage all of you to be active participants, ask questions, and share your insights with the experts. The success of this program will depend upon your engagement and dedication to the learning process. As well, I would like to express our sincere gratitude to the organizers, the dedicated team behind the scenes, and the esteemed speakers who have generously agreed to share their knowledge and expertise with us. In conclusion, so this National Faculty Development Program is not just an event, but an investment in your professional growth and the betterment of education as a whole. Okay? We all are excited to be a part on this journey with you, and we are confident that the knowledge and skills you gain here will have a lasting impact on your career and the students you influence. So let's make the most of this opportunity to enhance the faculty excellence, promote innovation, and contribute to the betterment of education. I wish you all a productive and enriching week ahead. Thank you for being a part of this program. Thank you.
Thank you, sir. Today we have our esteemed resource person, Dr. B. Reddy, sir, Director of Analytical R&D at Slayback Pharma, Hyderabad. Dr. Reddy, sir, brings a wealth of experience and expert facts to guide us effectively in our pursuit of faculty excellence, research competence, and innovation. I now request Mrs. E. Jazli to give a brief introduction about today's esteemed guest speaker, Dr. B. Reddy, sir.
Good afternoon, Amriti sir. So welcoming for this faculty development program, for sharing your thoughts. Thank you for accepting this.
Good afternoon, everyone, and welcome to our event. Myself, Jazli Eluru, working as Assistant Professor in the Department of Pharmaceutical Chemistry. It's my pleasure to introduce our today's guest speaker, Dr. Reddy. Dr. Reddy, who has graciously accepted our invitation to join us today. He is an accomplished professional with a strong background in analytical method development, validation, and method transfers in the CGMP regulated industries. With over 19 years of experience, Sir has consistently demonstrated expertise in various levels of pharmaceutical research and development, contributing to the successful creation and quality assurance of pharmaceutical products. Throughout his career, Sar has managed and coordinated activities across multiple geographies, including the USA, Canada, UK, Spain, France, and India, involving third-party CRO, CMO for product development, release, and stability testing of finished drug products. His extensive experience also includes troubleshooting technical challenges related to analytical activities in R&D, CRO, and CMO settings. A key aspect of Dr. Hanvi sir is expertise in preparing nitrosamine impurity risk assessment reports, developing and validating analytical methods using design of experiment principles under the design framework. His knowledge extends to various CMC activities, including extractable and leachable studies for ANDA, medical strictness. Dr. Hanvi sir has a strong command of regulatory guidelines, including CGMPs, CGLPs, ICH, USP, and FDA standards. And he has played a crucial and responding to analytical queries from regulatory authorities. Furthermore, Dr. Hanvi sir possesses excellent communication and interdisciplinary teamwork skills, having effectively interacted with researchers from diverse disciplines in a team environment. He has a commendable track record of making successful presentations in meetings and seminars, and he is recognized as a self-starter with the capability to write scientific and technical documents. In terms of dosage preview, Dr. Hanvi sir is expertise in preparing nitrosamine impurity risk assessment reports, developing and validating analytical methods using design of experiment principles under the design framework. His knowledge extends to various CMC activities, including extractable and leachable studies for ANDA, medical strictness. And he has played a crucial and responding to analytical queries from regulatory authorities. Furthermore, Dr. Hanvi sir possesses excellent communication and interdisciplinary teamwork skills, having effectively interacted with researchers from diverse disciplines in a team environment. He has a commendable track record of making successful presentations in meetings and seminars, and he is recognized as a self-starter with the capability to write scientific and technical documents. covers a wide range of pharmaceutical forms, including oral tablets,
3:30
hard and
computer lab
soft gelatin capsules, lipid solutions, parenteral, intramuscular formulations, and suppositories.
3:30
Dr. Hanmi sir has been recognized and awarded for his contributions, including for developing complex analytical methods at Dr. Reddy's laboratories, and appreciation from Slayback Pharma for achieving the first cycle approval of a complex ANDA product within a 8-month timeline. Sir is an inspiration for all of us. He was an assistant manager in 2011 at Dr. Reddy's laboratories and became a manager by 2016 at Celetrion, South Korea. Now he is serving as a director at Slayback Pharma. Hanmi Reddy sir holds a PhD in chemistry from JNTU in 2020. His research work has resulted in multiple research and review articles as well as several granted patents, showcasing their contributions to the pharmaceutical industry, scientific and technological advancement. Thank you, sir. I'm really grateful for this opportunity.
Thank you, madam. Hanmi Reddy sir, all the participants are eagerly waiting for your insights and guidance. So I request you to please take over the session.
Thank you everyone for attending this session. I express my heartfelt thanks to Sir C.R. Reddy College of Pharmaceutical Science and Sciences Organization, and my sincere thanks to correspondent Sri M. Rajendra Mohan Rao. My grateful thanks to Principal A. Jajuli and C.H. Amanya. And I just remember, like myself, Jajuli worked together in Dr. Reddy's, and she's we work for one of the product for US market. So she's also a quick learner and hard worker. So nice to meet you in this session. And I also thankful to all the participants to make this event successful. Can we start the presentation? Do we have a PPT with you, or shall I present it?
Sir, you can present, sir. You can present, sir.
Okay, yeah, thank you.
నేను నెట్ అయిపోతున్నాను సార్ మైక్రోసాఫ్ట్ టీమ్స్ నుంచి ఎందుకంటే నేను ఆల్రెడీ ఇక్కడే ఉన్నాను కదా నెట్ అయిపోతే మళ్ళీ ఫోన్ ఆఫ్
మొత్తం తీసే
నేను మొత్తానికి బ్యాక్ వచ్చేసాను జస్ట్ దీనికి కనెక్ట్ అయ్యి ఉన్నాను హాట్స్పాట్
సార్ మీరు కనెక్ట్ అవసరం ఎందుకంటే నాకు నెట్ ఉంచు.
ఓకే సార్.
Can you see my screen?
Yes, sir.
Is it visible in full screen?
Yes, sir. Yeah, we have to open it in full mode, sir. It is visible, sir.
Okay, it is in visible full view mode. Okay.
Sir, it's not in the presentation mode, sir. Just it is a PowerPoint mode.
Okay. Is it visible in PowerPoint
now
mode?
it's okay, sir. Now it's okay.
Yes.
So today we are going to discuss about nitrosamine impurities, which is very trending topic in the pharmaceutical industry. If you talk to any pharmaceutical industry person, nowadays they are worried about the nitrosamine impurity. Lot of regulatory questions on nitrosamine impurity. So today we are going to briefly discuss about nitrosamine impurities, what is its impact, how it will generate, what is its regulatory impacts on the current regulatory pharmaceutical industry. Today we are going to discuss following topics. It's introduction to the nitrosamines, how it forms, chemical background, what are the drugs recalled for this nitrosamine contamination, what is the regulatory overview on this nitrosamine impurities, analytical methods, what is its challenges, and how industries follows the risk assessments. If you see the structure, it starts with amine group attached to the NO. So the essential feature of nitroso compound is N connected with NO structure. So nitrosamine compounds are potent genotoxic agent in several animal species. Some are classified as possible human carcinogens. Some nitrosamine may increase risk of cancer if people are exposed to above acceptable level over the long period. High potency mutagenic carcinogenics like N-nitroso and alpha-azoic compounds are called as coherent of consent as per M7 guidance. Many N-nitrosamines are classified as probable carcinogens by IRAC. In a day-to-day life also, we also export to the nitrosamines. These nitrosamine compounds may found in water, smoked food, especially smoked food, cured meat products, processed fish, and other alcoholic beverages. So it's not, nitrosamine impurity is not due to human exposure, but in a pharmaceutical industry, when we are supplying a quality products, we should be ensuring that pharmaceutical products, the first prior important point is quality of the product. So when we are going to supply a quality of the product, it should be free from any of the impurities which can cause cancer and any other adverse effects. So these nitrosamines are it is observed in our day-to-day life. So we need to be very cautious when you are taking such kind of foods. So why nitrosamines are carcinogenic? So there is a article available in the literature. The genotoxicity of the nitrosamines depends on the alpha carbon hydroxylation. So if you look into the structure of nitrosamine, if it is having a alpha hydrogen, it is called alpha hydrogen. It oxidizes and forms in presence of cytochrome P450. It forms a hydroxy bond. Then it is converted as a imidazole ion. It can react with DNA and forms. It can create carcinogenicity. So different type of nitrosamines, whether it is cyclic ring or aliphatic, the effect is similar when it is having alpha hydrogen in nitrosamine impurity. So the term nitrosamine describes a class of compounds having chemical structure of nitroso group, NO bonded to the amine, nitroso amine. How it forms? Generally, nitrosamines forms when it is reacted secondary amine or tertiary amine, quaternary amine in presence of nitrous oxide. In acidic condition, it forms nitrosamine impurity. FDA identified several nitrosamine impurities that theoretically could form in drug products, and it published a guidance also in February 2021. If you take a step back here, you can see there are 2 points here we need to remember. The first thing is amine source. The second one is nitrosating reagent. So when these two chemicals react, it forms nitrosamine. So for amine source, there are multiple categories available, which are secondary amine. This is very prone to form nitrosamine. Tertiary amine, cyanamide, guanidine, amidine, hydrazines, hydrozones, hydroxides, hydroxylamine. So these are the source of amines which can form nitrosamine impurities. As well as nitrosating source. So standalone, if you have a amine source, nitrosamine may not form. It should have nitrosating source also. So these are the nitrosating source components which can react with the amine source and form nitrosamines. So generally nitrous acid and nitrate salts, nitrogen oxides, we classified as nitrosating source. In an industry, we we generally call with short names NDMA and the NDEA, which are called nitrosamine impurities. How this short name derived? If you look into the name of NDMA, N-nitroso dimethylamine, that means the amine group contains 2 methyl groups. This one. Whereas NDEA, N-nitroso diethylamine, it contains 2 ethylene groups to the amine group. This one. So there is similarly NMPA, N-nitroso methylphenylamine. That means methyl group, and whereas phenyl group attached to the amine source. So these are the different type of nitrosamines. We also call it as process related or small nitrosamines. As we discussed earlier, the secondary amine can form nitrosamines. This is the possible structure, possible reaction mechanism, how the nitrosamine amine forms. So nitrated and in nitrous acid, these are the source of nitrosating agent, which reacts with secondary amine and forms nitrosamines. Tertiary amines also forms when you look at the structure of nitrosamine. This is only a secondary amine. So wherever nitrosamine forms, there is a structure of secondary amine only. There is no tertiary amine or quaternary amine. Nitrosamine forms with secondary amine only. So if you have a secondary amine, it is possible to form a nitrosamine impurity. Whereas tertiary amines, it will form a dealkylation. It will lose one carbon group. Then it will form nitrosamine. So when nitrosamine forms with the tertiary amine means there is a dealkylation possibility. Then only it will form stable nitrosamines. Yeah, nitrosation of primary amines. So generally nitrosamine forms with the primary amines, may not be stable, and it will degrade further. So the primary amines not considered as a precursor of nitrosamines. In formulation, in pharmaceutical industry, there are many solvents we use for reaction synthesis as well as the product formulation. So one of the things are NMP, N-dimethyl formate. So these are, we call it as amide solvents, which are susceptible for degradation and can degrade to dimethylamine. When it is reactive with the sodium nitrate, it can form N-nitroso dimethylamine. So when API R&D person who is going to develop his synthesis process, he need to be ensure that what kind of solvent he should select to avoid the nitrogen formation. He has to take care what grade of the solvent he is using. If DMF solvent can create a secondary reaction of dimethylamine formation, he can avoid high reaction conditions and he can avoid nitrosating reagents. These are the general reactions where nitrosamine forms. As we discussed earlier, dimethyl formate can form a dimethylamine, which can react with the sodium nitrate, forms NDMA. Similarly, trimethylamine, it degrades diethylamine and reacts with the sodium nitrate and it forms NDEA, similar to the NMP. In a regulatory industry, the nitrosamine impurity first identified in 2018, and FDA has recalled certain products, that is valsartan product. After that, there are many products recalled from the market because of the over limit of NDMA impurity contamination. So many pharmaceutical companies, including Heterolab and other Aurobindo companies, they recalled their products because of the over limit, is more limits of nitrosamine impurities. You can see the progress of nitrosamine impurity identification over multiple products. In 2018, the first identification, followed by 2019, the other it is extended to other products, pioglitazone, losartan, and not only FDA, the other regulatory agency like European Medical Agency EMA and other agency regulatory agencies also, they recalled their product for contamination of nitrosamine impurities. So what are the drugs they recalled? One of the some of the products I listed here: valsartan, losartan, irbesartan. These are the sartan products where they having particular ring. To form this ring, they are going to follow particular route of reaction synthesis that can lead to the nitrosamine impurity formation. So in these products, you can also see ranitidine. Ranitidine is a very regularly used product in our daily life, but this also contains nitrosamine. You can see terminal tertiary amine group here. It can form nitrosamine. Even it is having secondary amine which can form nitrosamines. So these are the some of the molecules where FDA has recalled for nitrosamine contamination. So as earlier discussed, the process-related nitrosamines, NDMA, NDA, also called it as smaller nitrosamines, it is extended to the product-related nitrosamine that is called NDSRIs, N-nitrosamine drug substance-related impurities. Those are called NDSRI. So you can see in 2018 where it is contaminated only for NDMA. In current scenario, the product related means where product contains secondary amine or tertiary amine which can form nitrosamines. So now it is going to be a challenge to the pharmaceutical industry to control the nitrosamines in the product. So some of the examples I presented here. So if you see the dabigatran is blood thickener. We used it during the COVID period also. So it contains a secondary amine. It forms nitrosamine. So this is also one of the nitrosamine. So agency asks control those nitrosamine impurities before approving the product. Similarly, and quinapril, there are nitrosamine secondary amine source is available. It is forming nitrosamines. So since it is going to product patient safety issue, FDA has formed a nitrosamine task force team and getting the information across the industry, across the regulatory bodies to define certain guidance to the industry. So FDA also suggesting the pharmaceutical manufacturers how to mitigate the nitrosamine risk assessments. So one of the sources nitrate source. So agency is encouraging to use antioxidants which can treat as a nitrite scavengers. So if you use ascorbic acid or alpha tocopherol, this inhibits the formation of nitrosamine impurities. So agencies suggesting use antioxidants in the formulations to act as a nitrate scavengers. And if you see the earlier reaction mechanism, these nitrosamine impurity formation majorly in higher temperature as well as acidic condition. So in a formulation environment, nitrosamines may not form in basic or neutral environment. So when the formulation development, when we are going to develop the formulations, considering the environment of the formulation as a neutral or a basic to minimize the nitrosamine impurity formation. Like FDA has published some guidance in 2021, which contains 7 impurities, whereas Health Canada also published 18 nitrosamines in May 2022. And this Brazilian regulatory agency published 28 nitrosamine in June 2023. This is one important slide we can refer. We are considering nitrosamine as a high risk. We also need to see how it is mitigated as a high risk. Suppose we are we are going to consider the drug treatment as well as maximum daily dose, therapeutic indication, and maximum daily dose. So suppose if you are considering the dosage more than, suppose you are taking a drug more than a year, like Sartans, these drugs you are going to take it as for chronic usage. So it is like a more than 10 years if you are using, we need to consider it as a high risk products. If it is more than 1000 mg of maximum daily dose, those components we have to consider as a high risk. So thorough evaluation should be there for those products when we are going to release into the market. Whereas very low risk products, whereas below 1000 mg, and whereas it is going to use for only acute treatment, then it is considered as a very low risk. So based on the maximum daily dose, doses, therapeutic usage, duration, it is considered as a very high risk or low risk. FDA has given acceptance criteria for the nitrosamines. Look into the smaller nitrosamines, NDMA, 96 nanogram per day. This is the acceptance limit published in the FDA guidance. So these are very tighter limits. If you look into the maximum daily dose of a drug which is less than 880 mg, it should not cross more than 0.03 ppm. For drug product more than 880 mg, it should not cross more than 26.9 nanogram per day. That means very tighter limit. We in a pharmaceutical industry we consider it as very tighter limit. How this the acceptance calculated? ppm equal to A acceptable intake divided by maximum daily dose. In earlier slide, we discussed about the smaller nitrosamines. Now, FDA has published another guidance which is related to the NDSRI, that is product-related nitrosamines. In dabigatran, we discussed N-nitroso-dabigatran. So similarly, the product-related nitrosamines has classified into 5 categories. Category 1 given an acceptance limit of 26.5 nanogram. If you see the earlier slide, these are the smaller nitrosamines acceptable intake. This is 96 as well as 26.5. 26.5 is the smaller acceptance limit. So based on the data available across the industry, they classified potency category 1, 2, 3, 4, 5. So in category 1, considering as a 26.5 nanogram per day. Category 2, they consider as a 100 nanogram by considering NDMA as a reference. Category 4 considered as a 400 nanogram per day. Category 4 and 5 considering as a 1500. As per M7 guidance, the 1.5 microgram per day, it says also considered as a 1500 nanogram per day. So nitrosamine impurity, how this acceptance criteria has defined. As discussed in the earlier slide, the major problem with the nitrosamine is availability of alpha carbon, so that alpha hydrogen, which can form say hydroxyl group that can react with the DNA. So similarly, he has like FDA has given some guidance and flow charts how to define the potency category. One of the example is N-nitrosoketamine. The first step, does NDRSA have any hydrogen on its alpha carbon? If it is no, then we can consider straight away 1500 nanogram. If yes, we have to go to the next step. In the next step, okay, does any alpha carbon? Yes, alpha carbon is available here. Does NDRSA have any tertiary alpha tertiary alpha carbon? Yes. Hello. This is tertiary alpha carbon. So it is considered as a 1500 nanogram as per the guidance. So potency category 5. So this, so now industry having some guidance reference to define the limits for the nitrosamine impurity, particular to the drug substance related. So these are the some of the list FDA has listed in category 1, which is under 26.5 nanogram per day. You can see everywhere the nitrosamine forms with the secondary amine, and it is also having tertiary terminal methyl group. So in category 2 also, some of the products they have listed down. Just referred here for information purpose. Category 3, 400 nanogram per day. Category 4, 1500 nanogram per day. So category 5 also, it is 1500 nanogram per day. So now the real challenge to the analytical team to develop the analytical methods. Whereas if you look into the A limits for common nitrosamines, NDMA, as well as product related nitrosamines, we have very tighter limits, like in ppm level or ppb level based on the maximum dilute dose. So the real challenge for what should be the required sensitivity for the analytical methods. Generally, the nitrosamine impurity level should be less than acceptable intake limit. So if you take an example of valsartan, it is 320 mg per day. So 96 nanogram by 320, it is 0.3 ppm. So to develop a method with a point method is going to be a real challenge. So now the agency expectation is when you are going to develop the method, not only a detection limit, you need to have a quantification limits. That is limit of quantification. Limit quantification at that minimum level should be accurately quantified and precise, then considered as a limit of quantification. So the minimum requirement for this requirement is LOQ should be less than A limit to meet the regulatory requirements. When we are developing an analytical method for N-nitrosamine impurities, we need to consider the properties, physicochemical property of the N-nitrosamines. The majority of N-nitrosamine impurities reported found in pharmaceuticals are polar compounds. Is it ionizable? Yes, major are neutral. Some of them are basic. If you see here, this is the basic N-nitrosamines. This is acidic nitrosamines. So majority are neutral. Some nitrosamine may degrade at a high temperature. So while we developing method, we should avoid GC conditions if it is sensitive to the temperature. So nitrosamines solubility need to be considered. Again, that's going to be a challenge by considering the stability of the impurity. Again, the some of the nitrosamines are volatile, some of are semi-volatile. So developing a GC method with semi-volatile is a real challenge. So what are the techniques available for nitrosamine impurity quantification?
Slide number 46. 46 slides.
So detection and quantification of these traces nitrosamine in API and drug products can be a challenging and necessary use of advanced analytical techniques to meet the regulatory requirements. One of that GCMS, in case nitrosamines are volatile or semi-volatile and the API does not degrade at a high temperature, we need to use GCMS. LCMS, this is most predominantly using in industry. A broad range of nitrosamines, thermally liable APIs, which may form nitrosamine. So LCMS is majorly using in industry for nitrosamine impurity quantifications. Regulatory agencies publishing methods continuously to support the industries. Also encouraging industries and educational institutes to publish the analytical methods for nitrosamine. So this is one point I would like to emphasize here. When your researchers are focusing on the new publications or new techniques, this is one area you can look into and focus, because now nitrosamines, any paper. Sorry.
Hello. Madam.
Okay. So, sorry. Does anyone have a question? Okay. This is one area you can focus for. We encourage your researchers to work on the nitrosamine impurities. Now, this is agencies is looking for the support from the educational institutes, as well as even companies is funding for nitrosamine impurities method developments. So industry also follow the risk assessment practices. Somebody is not speaking. Can you keep it in mute?
Presentation more lessons.
Sir, please go ahead, sir.
Yeah. Okay. So, suppose nitrosamine is a big topic, but as well as the team has to understand how this forms, whether it is really a risk factor is available or not. So, assessing the possibility of having nitrosamine impurity in drug product, try to eliminate completely or control the permitted level. So this can be done in 3 steps. One is performing the assessment. Assess the risk of nitrosamine, potential contribution from all components of drug product. That is risk assessment. Once the risk assessment completed, if risk identified, then we need to do a confirmatory testing. If confirmatory testing results are below the AA limit, there is no. If it is above AA limit, then we need to provide a remediation controlling strategy that reducing the level of nitrosamine impurities. This is nitrosamine impurity risk assessment flow chart for in a drug product manufacturing industry. If you see the major, it will start with the API vendor, where API drug substance has to look into and route of synthesis process and raw materials. Next one is excipient. excipient water using in the drug product. So next, process. What kind of process we are using? Whether is there any cross contamination is possible in the process? Is packaging material? What kind of packaging material we are using? Whether it is possible any kind of nitrosating source? And during the stability, in the stability, is there any increase of nitrosomal impurities? So these are typical risk assessment program generally we follow in the pharmaceutical industry. So in a drug substance, as discussed, the starting material intermediate of the drug substance has to be evaluated thoroughly. What kind of solvents we are using? Is there any impurity that is having amine source, any nitrosating source availability, and what kind of reaction conditions we are using. So these all factors need to be considered for the risk assessment from the drug substance manufacturer. Second one, yeah, nitrate source. In a drug product, if nitrate source is not available, so even drug is contains a secondary amine, there is no, we can consider risk is nil. But when compared to the when considering the nitrate source from the excipient, there is a publication available where nitrate content has been established and tested multiple excipients, and you can see the number of formulations, number of nitrates. So all the excipients majorly using in the pharmaceutical industry contains nitrate content. So it is very much important to select the right grade of excipient in the formulation which contains less amount of nitrate. It can favors to the less controllable nitrosomal impurities. So this is risk assessment, whether the potential source. So we follow this kind of risk assessment flow chart. Similarly, like as discussed earlier, what kind of solvents we are using? Is it contains any amine source? Is the solvents are using? Is the solvents are recovered solvents? Is the water source contains any contamination? So all these points to be considered when we're doing a risk assessment from the API vendor as well as the drug product manufacturer. So we need to give the potential score for this. Yes, list of solvents used in the process. Is the chloramine is used in the water source? Is the potable water is recycled? So by considering all this, the risk factor score will come. Based on that, it's considered whether it is high risk or low risk or medium risk. And stability. During the stability during the process, is there any cross-contamination possibility while cleaning? So one of the factor is in an industry we use for multiple products in a same equipment. So when we are going to change the product, one product to another product, thorough cleaning is important to avoid any cross-contamination from one product to the another product. Again, the packaging material. So nitrocellulose is a polymer used binding agent in inks. So whenever there is a packing material comes, we need to evaluate that nitrocellulose is used or not. So if nitrocellulose, it can generate nitrites. That is a source of nitrosating agent that can form a nitrosamine impurity. So selection of packing material also important, and while considering the risk assessment, this also considered in the one of the factor. So these are the points just discussed in the nitrosamine impurity. If you guys have any questions, we can discuss.
Thank you, sir. So dear delegates, we are now open to queries. If you have any questions, please feel free to ask.
Sir, if the good afternoon, sir.
Yeah, good afternoon.
Sir, if the concentration of nitrosamine is more than what is required, can it prove to be carcinogenic also in the long run?
Yeah. See, if you see the A limit, which is described in the two FDA guidances, one is small nitrosamines, another one is product related. So the first target is we should be controlled below our AA limit. Okay, if it is crossing the AA limit, again we need to discuss with the agency that because if nitrosamine is above AA, we cannot directly release the material to the market. We need to inform to the agency, because if all the products are containing nitrosamines above AA, if the product is unable to release in market, agency also not going to have the product in the market, because there is a scarcity in the market, and product availability to the patient also will be difficult. So agency is taking a risk-based approach, whether is really what kind of product the supply is supplying. As discussed in the earlier slide, whether it is the duration of the treatment. If it is acute, no problem. They will allow into the market even if it is above AI. There is an example where agency has accepted because there is no supplier in the market, because to consider the patient safety, they released it. Because now this guidance M7 or this nitrosamine guidance, considering that the same it's considered probable carcinogenic agents, because it is exposed to the 70 years continuously. 1 lakh in 1,000, 1 in 1 lakh going to get cancer. That is the probability. So this is a very conventional or the very conventional approach agency is following, but it cannot be applied to all the products. So that's why when you have a product with above AA, we need to reach out to the agency and ask agencies guidance whether we can release into market or not.
Sir, we can also refer ICH guidelines for this, right?
Yeah, no. ICH guidance M7 we can refer, but for nitrosamine, we have a defined guidance in with respect to the FDA.
Okay, sir. Thank you, sir. Thanks a lot.
Thank you.
Sir, good afternoon, sir.
Yeah, good afternoon.
Sir, actually, it is a very elaborative talk. And you covered many of the things. Can I know, is there any synergistic action with any of these drugs taken other than the cancer drugs?
Sorry, I didn't get the question.
Any drugs will show. Can any drugs will show the synergistic action with the nitrosamine?
Yeah. Any drug. Yeah, and one more point. As per M7 guidance as well as the FDA guidance, these nitrosamines are not considered for the cancer drugs. Whereas, if you see the S9 guidance ICH, suppose if you are treating to a advanced cancer patients, we cannot consider this nitrosamine as, because already patient is in critical situation, and that is only for one-time usage or very short-term usage. This nitrosamine may not consider as a risk, and this nitrosamine risk is not considering for the advanced cancer drugs. Only our regular products, which are especially chronic use.
And one more thing, you have covered your about packing. Is there any storage limitations for this drug? Because it may degrade, right? So that's why. Is there any storage limitations for this type of drugs?
Storage. Storage means for any drug product or drug substance, there are stability studies going to perform. Based on the stability and when we are performing development stage stability studies, if it is meeting the product specification, then we are going to define the storage conditions. So as you know, 25 degree, 40 degree, and refrigerated condition 2 to 8 degree. So suppose if product is not stable at 25 degree, then we are going to recommend the refrigerated condition 2 to 8. So all depends on your developmental data, where you have a sufficient data to define your storage conditions. So
thank you, sir.
With respect to your question, there is no specific recommendation to the storage, because in long term in accelerated conditions, that is 40-75 conditions, where it will accelerate the especially valsartan case. During the storage, the limits, the impurity content is increased in valsartan case. So in storage, in the long-term storage or in the accelerated condition, nitrogen impurities are increasing. Means that is favorable to that condition. So it may not be applicable. It may not be common for all the drug products. In particular products, this mechanism may happen. But again, it's case-to-case study. We need to evaluate.
Okay, thank you.
Reddy sir, I have one question for you.
Yeah.
Can you hear me, Dr.
Yeah.
Sir? Yeah.
Yes, Dr. Sivaram.
Good afternoon, and really, I really appreciate for your good presentation, for giving out the topic on this nitrogen impurities. I have one question. Generally, in pharmaceutical industry, when the finished product is being developed, we carry out tests for impurities, isn't it?
Yeah.
And generally, the value should be always less than 1%. At that time, do you think that these kind of problems might have not been evolved? That's one question.
Yeah.
And the other question is, how the problem has arisen? Is it from the clinical trial studies or from the adverse drug reaction that has been obtained in the general usage of the patients?
Yeah. Okay.
That's the second question I want to ask.
Yeah. Thank you, Dr. Shivam. Interesting questions. Thanks for raising this question. Yeah, the first question. Yeah, as you rightly said, impurity evaluation or estimation controlling in pharmaceutical QC lab is correct. But these are the impurity. See, nitrosamines. When with respect to the product quality, we always consider the possible impurity which can form along with the impurity with the drug substance manufacturer, as well as the storage, or we also do a forcible degradations. We add, we keep it in oven for some time, and we add forcibly acid or base to understand the degradation pathways, and considering all those impurities for our product specification. Means these are the impurities we are going to get during this product stability. So those impurity we are going to monitor it. But nitrosamine impurities are not generally, if you see the ISH guidance Q3, 2 and where there is a general guidance for drug substance and drug product, which talks about the impurities in percentage level, mg level. We can consider mg level. Okay, 0.1%, 0.2% or 1%. Okay, based on the maximum daily dose of drug. So these impurities, we are talking about nitrosamine impurities in nanogram level. So even though those impurities are present in our earlier product, ranitidine, we are taking the last 10 years we are taking the but people are not reported with that impurity because it is not detected in our regular HPLC method. Okay, now agency has, because nitrosamine is not a new topic. It's not only present in our pharmaceutical products. As discussed in the presentation, it is there in your smoked food. If you take continuously weekly smoked food, it is possible get a cancer. Okay, so so nitrosamine impurity is discussed in many ways. One is drinking water. Whatever the water we are drinking, if you test for nitrosamines, NDMA may be possible, some ppm level. If you are taking continuously, there is a chance. Okay, so since pharmaceutical products we are supplying to a patient, and that should have a control. So this nitrosamine is also coming to the control, especially the valsartan. If you see a valsartan product continuously more than 10 years, one patient going to take. So when you give valsartan to continuously one person more than 10 years, how much amount of nitrosamine it is accumulated exposure to be there. So that's why agency has come into conclusion that yes, we need to control the acceptable intake of nitrosamine with respect to all the products going to supply to the market. Hope I answered your two questions.
Very good. Thank you very much for clarification.
Yeah, thank you.
Sir, good afternoon, sir.
Thank you, Dr. Reddy, for clarifying me the questions. Really appreciate for your comments.
Working as assistant professor in the Department of Pharmaceutical Analysis. Sir, I have small doubt in the formation of nitrosoureas. Which one is more stable in between both primary, secondary, tertiary amines?
Secondary amine nitrosamine is more stable.
Okay.
Secondary amine nitrosamine is more stable.
Okay, thank you so much, sir. Very informative session, sir. Thank you so much.
Yeah, thank you.
Madam, please unmute yourself.
Good afternoon, sir.
Yeah, good afternoon.
Hello, sir. This is Dr. V. Anita Kumari, sir. A small clarification, sir. Sir, there may be any chances of drug interaction, sir? So because you told to reduce the oxidation, antioxidants also can be combined in order to reduce the unwanted effects. So is there any chance of drug interaction, sir, with antioxidants? For example, tocopherol and other classes of drugs are there. There may be any chances, sir, how to control those types of unwanted effects, sir?
Yeah, good question. Yeah, see, it is one of the suggestion. It is one of the suggestion. Okay, but again, we need to see the impact of antioxidant. Okay, generally antioxidants are recommended to use to certain level. Okay, what kind of doses from using that with a certain level only it is recommended. Again, we need to do check our excipient database, which is available in the FDA website, where maximum usage of that particular excipient for the particular formulation. Even the route of administration also, even oral injectable products, each excipient having particular exposure limit also is defined in the database. So for each excipient we need to use. So when come to the drug excipient interactions, yes, I do agree, we need to study that. Generally the antioxidant why we use to minimize the oxidation impact or to minimize the degradation. So using the antioxidant may not impact because it is going to help the product to minimize the degradation. And one more thing, the antioxidant also having the acceptance limit with respect to the database.
Okay. So there may be chances of diverse group of functional groups, na, sir. Antioxidants are not having common functional groups, sir. They are having diverse group of functional moieties in their chemical structures, na, sir.
Correct.
That is my doubt. Just small.
Yeah. If you see the presentation where it is talks about the particular publication is available where it talks about even share after this session also the effect of antioxidant to control the nitrosamines. So there is a publication I can share you. So you can see why they listed only tocopherol and impurity tocopherol, because that is having more impact to control the nitrosamines.
Like, comment.
One more, sir. One more question, sir.
Yeah, yeah.
There are any specific identification tests, sir, for the nitrosoureas impurities in the raw materials or finished goods, whatever it may be in process materials? Any specific identity tests, sir?
Okay. See, first point, identity test means
identification test.
Identification. See, first of all, it's going to be very expensive test to have identification for each raw material. So that's why in industry we do a risk assessment. If risk assessment is not in risk assessment, we found it is not possible, then we will not do any testing. If risk assessment says yes, possible, then we are going to incorporate even not only identification, we are going to do a quantification test also for particular nitrosamines.
Sir, for each compound, each drug product in the industry, all the raw materials are must and should be properly monitored, sir. Monitored, they should be analyzed, sir. After the test pass only, it is approved for the in process.
Correct.
When it is when the drug that impurity is present in the raw material, how can we monitor the situation, sir?
Okay.
Yeah, there are any monitoring steps, sir?
Good question here. One more good question from you. Here, now the nitrosamine impurities in 2018 started with one product. It is extended to the other products. Okay. Whereas for drug product related or the common nitrosamines is evaluated now. Okay. So even now only drug product manufacturers are focusing. Okay. Now it is extended to the suppliers, even the API vendors, even the raw material suppliers, even the excipient suppliers also. Now they are going to focus on the nitrate source content, as we discussed in the earlier nitrate source, because they also need to do a nitrosamine risk assessment as a supplier. If they say that yes, nitrosamine contamination is not possible, we no need to do that particular testing. If they say they need to control and we need to incorporate the testing. Okay, so for each raw material or each material, we need to have a particular declaration from the vendor.
Yes. So the label must be there in order to identify. So label conditions also must be stated clearly in order to avoid the unwanted effects. So special instructions also can be given, suggested by the manufacturer or supplier, whoever it may be the party.
Yeah.
Okay, sir. So, thank you, sir. Thank you for.
Yeah. Particularly, there is a particular controls specification will be there. With respect to specification, we do a testing before it entering into the quality control lab or for release for the manufacturing. Okay. Apart from that, there are standalone declarations for elemental impurity, residual solvents, and nitrosamine. For these also, we are going to get a declarations or the risk assessment from the each supplier. Once we evaluate, we clear it, then only it is moved to the QC for release as well as the production.
Okay. Thank you, sir. Thank you for valuable suggestions and clarifications. Thank you.
Yeah.
Any more queries? I think there are no more queries. You clarified everything, sir. Thank you, Hanmi Reddy, sir, for an enlightening and thought-provoking session. Addressing nitrosamine impurities in this present scenario is crucial in case of health and safety concerns. Your patient and considerate answers to the questions were highly commendable, sir. We are truly privileged to have this opportunity to learn from your extensive experience. Your session has sparked curiosity and ignited a desire for further exploration, sir. Thank you once again, sir, for sparing your valuable time.
Yeah, thank you very much. And I expect I had a lot of good questions from all across the participants. Yeah, thank you very much for